Communications. Cells communicate through a signal transduction pathway where a ligand and receptor form a complex to create a cellular response. There are four methods of communication: direct contact, endocrine, synaptic, and paracrine signaling. Direct contact signaling occurs when cells associate plasma membranes and exchange ligand molecules. An example of communication is through gap junctions. Endocrine signaling occurs when a signaling cell targets a distant cell through the bloodstream and produces a long-lasting response. Hydrophobic ligand can penetrate through the plasma membrane. Hormones are present in small concentrations in the blood. Synaptic signaling happens when ligands are released from a signaling cell into the extracellular fluid of a narrow space, or a synapse, and the ligand diffused into the extracellular matrix of the other cell, or the ligand will quickly disintegrate. Paracrine signaling happens locally between cells and is short-lived because the ligand disintegrates in the extracellular fluid quickly and has a short-lived effect. Intracellular receptors are receptors located inside the cytoplasm. An example of an intracellular receptor is the steroid hormone complex. Hydrophobic ligand, or hormones, penetrate through the plasma membrane and unite with the receptor protein inside the cell, forming a complex. The complex then travels to the nucleus and acts as a transcription factor and binds to specific nucleotides, controlling gene expression. Another class of receptors are membrane proteins. A ligand-gated ion channel is a transmembrane protein receptor which opens to allow ions to pass in both directions only in response to the binding of a ligand. When a ligand-receptor complex forms, it creates a signal transduction pathway which causes a cellular response, opening the channel. A G protein is a protein that has 3 subunits and is activated by a ligand-receptor complex. When the G protein attaches to the ligand-receptor complex, GTP phosphorylates the G protein, causing the first subunit to dissociate from the other 2 subunits. These 2 subunits can activate the other membrane proteins. The G protein is restored when the ligand is removed. An enzymatic receptor is a transmembrane protein where the binding of an extracellular ligand causes enzymatic activity in the protein, producing a product, which is a cellular response. Phosphates play a role in signal amplification. Protein kinase is an enzyme that adds phosphate groups to proteins from ATP. Phosphates are proteins that remove phosphate groups from proteins. Cascade chains are proteins that phosphorylate one another to activate one another. The purpose is to amplify a signal, or to produce a larger cellular response. First, a ligand binds to a membrane receptor and produces an active relay molecule and activates kinase 1. Active kinase 1 transfers a phosphate group from ATP to kinase 2 to activate it. Then, active kinase 2 transfers one phosphate group from ATP to activate a protein. The activated protein produces a cellular response.